Thanks very much. So I was really excited to come here. I'm so excited to come here because I think all of you who are from the network science field have a lot of work to do with the C-elegans connectome. And you already saw Eduardo's talk earlier. I was so excited to come here that I wanted to show this audience all the cool things that are in the space. And as it turns out, there's way too many cool things to show in this space than I have time to show you. So you'll have to forgive me if there's things that I don't get a chance to talk about. Maybe we can talk about more towards the end. But if you saw Eduardo's talk, you got a lot of the basic intro. He did a lot of work for me in terms of introducing the C. elegans, the worm, and why it's important, all that good stuff. So let's just say he and I are very much fellow travelers along the path. I represent a group called Open Worm that's an international collaboration where we make many collaborations with different labs that are doing work in this space. And so his work is really an excellent leading light in this space. I'm going to try to say some things that are a bit different from what he said, just because I want you to see even more of this landscape. But one thing that we have in common is this concept of creating a full loop simulation with a body model and a nervous system, because the body model actually provides a really important vehicle for understanding how the dynamics of this particular complex network works. So I'm going to say, so my roadmap here for today is I'm going to tell you a little bit about what we've done in Open Worm to build a very detailed model of the worm body. I'm going to walk through a little bit of connectomics of the worm nervous system structure. Then I really want to kind of emphasize a little bit more of the complexity of the C. elegans nervous system than we commonly hear about, because I think it's super interesting, and I think the work of getting even better models of the nervous system, deeper models, is really fun and interesting. And hopefully if I have time, I'm going to get to talk about some dynamics and modeling that we do with the dynamics of this network, and then talk about some future questions. Okay, so we take inspiration for building our body model from industrial design type software that models the physical nature of devices, vehicles, like in this case the Boeing 777, down here a motor for a plane. Right, so we now have technology that allows us to not just make pretty visual animations, but actually simulate things down to the mechanics of devices. So it turns out that a large part of the C. elegans biology field has actually done a lot of biomechanical studies on the worm to understand things like its frictional forces when the worm is crawling around on a dish, the elasticity of the worm itself as you bend it. So experiments like this, you take a worm and you stick it into a pipette and you kind of bend it around to understand how internally elastic it is, or experiments like this where you're measuring how the body of the worm crawls, say on different viscosities of liquid. So there's a lot of this data, and so it would appear that it's possible to draw a line between software that can model complex devices and worms, even though it's still not been done and it's still rather complicated. So there's been quite a history of folks doing body models. Eduardo told you about one that he's been using, which is, I think, the third from the bottom there. And he's also been augmenting these models. So we've, in Open Worm, we've been working with a group in Novosibirsk that has been building a model that sort of goes by the tag name Cybernetic. And I want to show you a little bit about it. So we also have, we map the muscles of the C. elegans worm one-to-one into a particle-based simulation. And because I don't have a lot of time, I'm just going to jump to showing you some of the cool pictures. So on the left-hand side is sort of the behavior of a real worm being tracked. And then these two are generated images of what cybernetic looks like when it's running. So in this case, you're seeing the worm is crawling on a lawn of essentially an agar gel. And what's making it move is the contraction of the simulated muscles that are inside of its body. And of course, what makes those muscles contract is the dynamics of a complex network. So we've done some quantifications of comparing, swimming and crawling behaviors of real worms to what comes out of our model. We can generate curvature plots like this, which will become important later. And we can even do fun things like, in this example, the worm goes from a lawn of agar gel, where he's been crawling, and forgive the lower resolution of this, and then he sort of dives into this area over here, which is actually a liquid, and you can see him start to swim. So he's actually kind of fallen into the liquid, and now he's submersed in the water, and he's sort of muddying up the water there as he's swimming in that direction. So this is cool. So we have something that can give us a lot of one-to-one reality with the real organism, which then allows us to run all sorts of interesting experiments and compare it to real experimental setups. So, okay, but that body model is really just a vehicle for a nervous system. So let's talk a little bit about the nervous system. One of the things that we did several years back is to plot the C. elegans connectome using high plots. I'm not sure how many of you in this field are familiar with this way of visualizing, but very quickly you take the nodes in your graph, you divide them up into 3 categories, like in this case, regulator, manager, workhorse. Then you plot the degree of those nodes, and you put nodes on an axis based on their degree. And then you turn that into a rotational plot. So you can sort of see in rotation the relationship of this. So this graph is this graph here. If that went a little too quickly, forgive me. So there have been a lot of explorations of just the graph structure of the C. elegans network. Papers like this, which analyzed it, this graph node structure. So that's been available for quite a while. So we decided to play around with some open source tools that let us generate these hive plots, this graphing approach. And in this case, we put sensory neurons on one axis, motor neurons on another axis, and then we split the interneuron axis this way, so you can kind of see not only the relationship of sensory neurons to interneurons and interneurons to motor neurons, but also interneurons to interneurons because they connect with each other. And you can plot the chemical synapses, and you can plot the gap junctions, which are different kinds of connections in this network. And one of the things that kind of pops out when doing this is that you can see sort of this structure where up at the top of the interneurons there's a whole lot of connections that are kind of flowing into these sort of top guys up here. So we quantified this a while back. We looked at the top neurons that are in the space to see if in fact there was what's known as this sort of rich blood phenomena, which is something that's been observed in cortical networks of mammalian brains or human brains. I didn't see that having been shown yet in any of the topological analyses of C. elegans. So you can actually show that, yeah, the neurons that have the highest degree have statistically a much, much higher degree than you could predict just by going kind of by average degree. So there's some interesting structure in this nervous system, right? And so this is the kind of exploration that you can do with this network. By the way, this is available online on GitHub in an IPython notebook. We're very friendly to that kind of approach. But I want to leave you here with this idea of like, well, what is really under the hood there, right? When we draw these sort of ball and stick type diagrams. And to understand why the C. elegans nervous system is the only one that we've mapped out in an organism, it's important to understand the heroic work that went into producing that graph. Because it came from somebody slicing up the worm very finely, putting those slices under an electron microscope, and essentially following every single neuron through what is a massively complex soup. And the soup kind of looks like this. So when you see a chemical synapse, like what's a pretty little line with an edge, right, in one of those graphs, this is the kind of picture that you had to extract that from. This is an electron micrograph. If you take the outlines of this, the boundaries of this is a cell in slice that you've cut open, and you have to kind of identify that, oh yeah, this part over here looks like a chemical synapse based on my mental model of what a chemical synapse looks like. Similarly, this is what a gap junction looks like. Again, it would look like an edge on your graph, but the neuroanatomist has to look at it like this. And this is a nice three-dimensional picture that kind of shows you what those electrical synapses look like. So this is the entire worm if you were to slice it and you were to cut right through it. Okay, this is the soup again that you start with, and so you typically have to zoom in as far as like this little picture here until you can even start to see. And this is some of the original plates from that study. Like this is cell number 70, and then they determined cell number 70 is connected to cell number 71 because we can see that there's a connection right there. Okay, so why is this important? When you go through and you outline this and you see all these cells and you start making all these connections, you might get a picture that looks like this. Again, drilling in, it's like that complicated. And then, graphs like this were produced in this paper, and this looks like a familiar network graph, I think, from things that you might be familiar with. But it's important to know that, like, these cells are not little points that just sit in one place. They travel along the body of the worm, and they have very complicated connections. So So in this case, let's look at just one neuron. Okay, this is up at the front, the head of the worm. This is the mouth of the worm, and this is its head. And this is something that's called the nerve ring. And so this is one neuron that sort of starts here and curves around the nerve ring, and it goes down the end of the worm in this way, right? So this is a neuron called AVAL. That's the name of it. And so if you take a sort of a flattening approach to looking at it, to looking at the nerve ring, you might get a picture like this. And so this is what I kind of want to show you, that when this original connectome was figured out, these were the kinds of pictures that led to these graphs. So we're looking at one neuron now, which curves around the nerve ring, okay? So this is now kind of looking at the head and then seeing the neuron as it goes down the end of the worm. So end of the worm down here, head of the worm here, this is that neuron as it curves around. And I just want to show you really quickly, if you zoom into these three little areas here, okay, in that order, okay, this is where the other neurons are connecting. So all these letters here are the names of other neurons and they physically touched at that point of this cell, whereas other neurons were further down the neck of the cell, and they touched down here. And then yet other neurons touched further down here, right? So what I'm trying to show you, right, is that in the case of a real biological organism, to say that every single, to say that all edges are coming from the same place may not be the best, most exact model in this case. And so in computational neuroscience, there's a practice of taking a neuron and carving it up into different compartments and assigning synapses to different compartments. So if you think about this network, which seems very nice and friendly as 302 nodes, if you start to think about where edges actually hit, and in more complex models, you do this, you might actually break down a single neuron into, say, 10 compartments or 20 compartments, and then have edges going between all of those. So you might explode this network out into something that looks more like 3,000 nodes or further as you go. And in fact, this is what we've done in the Open Worm project. When we've reproduced the C. elegans connectome, we've done it in a way where we've taken the anatomy of these individual cells, turned them into a multi-compartmental description, and then, and yet still the work actually of assigning the positions of the synapses has yet to be done, but this allows us to do that. So this is a 3D visualization of looking at the more complex connection graph that you actually find inside of this network. So if there's one thing I want you to leave here with is that actually going down to this level of modeling the C. elegans graph and the C. elegans network may actually be quite valuable. There's other evidence from the dynamics of this nervous system that supports that as well. Okay, so I sort of promised I had a lot to talk about, and I know I have already not enough time to really tell you very much about dynamics, but one idea, I'm just going to kind of do a scattering of ideas, I guess, but in the 2 minutes that I have left. But optical imaging is transforming the way that we understand the dynamics of this nervous system. So in this case, we're looking at a fluorescent marker inside the nose of the worm again, as it's being stimulated by a chemical signal. And so if you do this across all of the neurons in the worm, because its body is transparent, you can start to generate pictures that look like this, with raster plots that are taken from now hundreds of neurons at a time. And again, this is the only animal that you can really look at almost the entire nervous system and see the dynamics of each individual neuron within an entire organism that's alive, that's behaving. This is still not true for the mouse and for the fly. So the work that we're doing in modeling, because I don't have too much time to talk about it, is reproducing these dynamics using the nervous system model that I showed you, and with the connectivity. So this is okay. Yep. So that's a reproduction that's beginning to qualitatively start to look like these kinds of rasters. We are making a lot of effort to try and reproduce, to make our nervous system produce oscillations that can allow for the movement that we see in our body model. I should point out that in this case, the body model is not being driven by the nervous system. It's being, very simple oscillations are being put into it, which are sufficient to produce this behavior. And so now we have a target for the output that we want our nervous system model to make. So we're using machine learning, some similar techniques to what Eduardo showed you, and but we're kind of putting our own flavor on it. This is also collaboration with other folks as well. Okay, so now it's time to show you that stuff. So hopefully what you got out of this was that detailed worm body simulation in 3D is helping us to create a vehicle that helps us understand the dynamics of this nervous system network. I showed you a little bit about what you can do with a simple graph analysis using some of our open source tools, and showing maybe a rich blood structure that's of interest in this space. I hope that you got a little bit about how this structure is more complex than is often discussed. I didn't get a lot of time to talk about dynamics, but what I hope you will come to appreciate is that the next 5 years of data that are going to be coming out in C. elegans are very exciting because of techniques like this optical imaging. And so I think if you're looking for like a biological space to break in network science, I would really recommend looking more at C. elegans. We're an international collaboration, and we have lots and lots of folks to thank, not only these folks, but also dozens and dozens of open source volunteers over the last 5 or 6 years. So we'll stop there and stand up for the questions.